Good day, class. In this episode, we are going to talk about translation. In the previous meetings, we talked about DNA replication and DNA transcription. Now, we are going to talk about the final step of central dogma, which is mRNA translation. Please take note that not all RNAs, which are produced by transcription, are translated. Remember that there are many types of RNA other than mRNA. For example, tRNA, snRNA, microRNA. These RNAs are coded by genes that we call non-protein coding genes. And when we express non-protein coding genes, we stop at the RNA level. We no longer proceed to the polypeptide level. But as far as mRNA is concerned, then we have to talk about translation because it is the mRNA, the type of RNA that is being translated to polypeptides. I am showing you now this picture once again for you to be reminded of the overview or for us to have a bird's eye view on where translation sits in the entire system. So focus your attention now on in prokaryotes. Notice that the prokaryotes don't have nucleus. So the moment the mRNA is produced, it can already be used right away for translation, for polypeptide production. But in eukaryotes, since we have a nucleus, then the RNA transcript that is directly produced by transcription is not yet ripe for translation. That RNA is yet to undergo post-transcriptional modification, which I left to you as your reading assignment. But medyo nag quick discussion ko gamay ani, but please read on your own about post-transcriptional modification. Anyway, the RNA transcript, the primary transcript RNA, that's how we call it, or the heterogeneous nuclear RNA is going to undergo post-transcriptional modification to produce the mature mRNA, the one that will exit the nucleus via the nuclear pores, which will then enter the cytoplasm, where it will associate with ribosomes for protein production. Okay? In any given eukaryotic species, the transcription of the thousands of different genes that an organism possesses requires appropriate timing and coordination. In connection to that, the main function of the genetic material is to encode the production of cellular proteins in the correct cell at the proper time and in suitable amounts. The different cells of our body produce different kinds of proteins, and it is using those various proteins that our cells are able to do their respective functions. Nasabi ata sa biochemistry that proteins are the labor force of the cell. They are the ones that do the functions of the cell. But remember that cells are only able to produce those proteins in the first place because they carry within them, within their nuclei, the genetic material, which is DNA. However, they have to express those genes at the proper time. I believe you know about stem cells. For example, there is a stem cell that is destined to produce a skeletal muscle cell. The skeletal muscle cell within it, mayroong mga myosin and actin filaments which are proteins. But kung stem cell pa siya, hindi pa siya nag-differentiate into a mature muscle cell. Syempre, wala pa siya masyadong mga myosin and actin. But the genetic material, the instruction needed to produce myosin and actin is already encoded in the DNA of the stem cell. Hindi lang nasa DNA ng skeletal muscle cell which is mature, but also in the DNA of the stem cell. Pero, yun nga lang, hindi pa siya na-express. Yung mga gene for myosin and actin, hindi pa na-express kung stem cell pa siya. So, ibig sabihin, ang point ko lang dito is genes are expressed at the proper time and also in suitable amounts. Nasabi ko na din to before ata na before our cells express the genes that they have, they first need to be induced. The key term here is induction. Dili ka na sila basta-basta magbuot-buot na mag-decide na i-express nila ang kanilang mga set of genes to produce these set of proteins. First, they have to be induced. They have to receive signals from the environment. Those signals can be in the form of hormones, cytokines, cytokines, for example, which you will learn more about when you reach a third year, when you discuss immunology. So, ayun, mga cytokines and hormones, those are examples of signals from the environment that trigger our cells to express certain genes. For your reading assignment, I will request you to read on Archibald Garrod and Alkaptonuria, and also Biddle and Tatum and their experiment with Neurospora crassa, which they used so that they came up with the hypothesis that we call one gene one enzyme hypothesis. So for the interest of time, I will not anymore discuss their experiments, but you can read them in the book. They are found in the book. I also included the page numbers at the end of my PowerPoint for you to easily locate the material that you have to read. So, ang i-explain ko na lang sa part nila Biddle and Tatum ay yung mga exceptions sa one gene, one enzyme hypothesis. Kasi, that hypothesis kasi has some weaknesses, and that's what I'm going to point out now. One weakness of the one gene, one enzyme hypothesis is that it assumed that all genes code for enzymes. The truth is, hindi naman lahat ng mga polypeptides na napoproduce sa translation ay ginagamit para makabuo ng enzymes. All proteins are encoded by genes and many of them do not function as enzymes. For example, ito sa picture, in the plasma membrane, you have your integral proteins and peripheral proteins. Some of these proteins act as receptors, others act as channels or transporters. So, di ba, hindi naman sila enzymes. Another example, hemoglobin. Ang function naman ng hemoglobin is to carry oxygen in the blood, not to catalyze reactions. Another weakness of the one gene, one enzyme hypothesis is that it assumed that only one single gene is needed to produce a single protein. But in truth, some proteins need more than one gene to be produced. For example, hemoglobin, mayroon siyang dalawang beta chain at dalawang alpha chain. Yung dalawang alpha chain, they are produced from one gene. But yung dalawang beta chain naman ng hemoglobin, they are produced by another gene. So, ibig sabihin, there are at least two genes that you need to express to produce hemoglobin. On top of that, iba pa din yung mga enzyme na kailangan mo to produce the heme component of hemoglobin, which you will talk about when you reach hematology. And those enzymes needed to produce the heme are encoded by yet another set of genes. So the main point here is to produce a single protein, most of the time, hindi isang gene lang yung kailangang i-express, but a multitude of genes. Isa pang weakness ng one gene, one enzyme hypothesis is it assumed that each gene can produce only one type of polypeptide. But of course, now we know that a single gene can produce different kinds of polypeptide because the pre-mRNA that is synthesized during transcription can be cleaved in different manners. I'm talking about alternative splicing so that several different mRNAs, mga mature na mRNA, can actually be produced from just a single gene. And following the logic, so ibig sabihin, several polypeptides din ang kaya nating i-produce using just one gene. And finally, ngayon, alam na din natin na not all genes code for polypeptides. Kung mayroon tayong tinatawag na protein coding genes, mayroon ding mga non-protein coding genes. Non-protein coding genes are the genes that, yes, they are transcribed into RNA, but the RNA produced is not mRNA. For example, tRNA, snRNA, um, miRNA, those RNAs are not translated. Instead, they are ends in themselves. Yung mRNA kasi hindi naman siya final product, di ba? It is only a tool to produce the final product, which is yung polypeptide. But itong mga RNA na pinagsasabi ko, itong mga tRNA, they are final products in themselves. tRNA in itself is already functional, and yung function niya nga is to deliver amino acids to the ribosome during translation. We will talk about that later. So, yun yung mga kailangang i-revise o yun yung mga weaknesses ng one gene, one enzyme hypothesis. Nevertheless, despite the weaknesses of one gene, one enzyme hypothesis, it has become an important milestone for our understanding of how proteins have a genetic basis. Now, let us talk about the structure of our mRNA. The mRNA contains a series of codons. Codons are groups of three nucleotides that most of the time code for a particular amino acid. Tinawag siyang codon kasi tingnan mo naman, if you look at the series of nucleotides in the mRNA, mukha siyang password. Mukha siyang um mukha siyang code talaga, like the Hammurabi's code. It's like something na mahirap i-discern, but actually if you know the pattern, and if you know the kumbaga, if you know what each codon signifies, then you will be able to understand the message, kumbaga, that is embedded in the mRNA. And because of that, we have this thing called genetic code, which is elucidated by Marshall Nirenberg. I am presenting to you this magnificent table. You don't have to memorize this table for the quiz and for the exam. I think you will be provided with this table naman, but familiarize yourself na lang on how you can navigate this table. You have 4 nucleotides forming your mRNA, but you only use 3 at a time to form a codon. 4 raised to the third power equals 64. So behold the 64 permutations of your 4 nucleotides. Out of these 64, only 61 code for amino acids, and these 61 codons are what we call the sense codons. Sa 61 na sense codons na iyon, may isa na napaka-espesyal, the AUG. We call that the start codon. The AUG codon codes for the amino acid methionine. That start codon is special because during translation, yung start codon na yan ang siyang hahanapin ng ating mga ribosome. Now, pag umabot na yung ribosome sa start codon, then malalaman niya na, oh, ito na pala yung dapat na pinakauna kong ita-translate. So, ayun, ita-translate niya na ngayon yung AUG into methionine. Sa 64 na codons, tatlo naman ang tinatawag natin na stop codons, termination codons, or nonsense codons. These codons do not code for any amino acid. Later, during translation, you will see that the moment na yung ribosome ay umabot na siya sa isa sa tatlong start code ay umabot na siya sa isa sa tatlong stop codons na ito, mayroong papasok sa ribosome, imbes na tRNA that carries one amino acid, iba yung papasok. And what is that? We call that the release factor. But more on that later. Now, let's go back to this diagram. Again, in the mRNA, you can see a series of codons that code for amino acids. Sila yung tinatawag natin na sense codons. In the very first part of the series, you can see a start codon, which codes for methionine. And at the other end of the series, you can see a stop codon, termination codon, or nonsense codon. In this diagram, it is UAA, but it can also be UAG or UGA. Now, another thing na dapat i-mention is not all parts of the mRNA is translated into a sequence of amino acids. Notice in the diagram, yung naka-green na rectangles, meron tayong tinatawag na 5' UTR and 3' UTR. UTR stands for untranslated region. So nasa pangalan na ng structure, ma-denote mo na kung ano yung characteristic niya. So it is a region of the mRNA that is left untranslated. Again, the 5' UTR and 3' UTR. Notice the length of the amino acid sequence dyan sa baba. Notice na yung length niya is naka-correspond lang sa length encompassed by the AUG up to the last sense codon before the stop codon. I hope you are getting my point. Ano lang, ang point ko lang is i-compare nyo yung length ng mRNA sa length ng polypeptide, 'di ba? Yung starting point ng polypeptide ay nasa AUG, nag-end siya sa codon na GAA. Hindi siya nag-end sa UAA kasi nga yung UAA does not code for any amino acid. Now, the the amino acid sequence did not begin from the 5' UTR. Kasi nga yung 5' UTR na yon is not translated. Again, translation begins only in the AUG codon. Now, let us see how mRNA associates with the ribosome. Diba, the ribosome, we know it is the machinery that synthesizes protein. So, the ribosome scans or reads the mRNA in the 5' to 3' direction. I would like to emphasize this class kasi mayroong notable na difference yung pagbasa ng ribosome sa mRNA. Diba, during DNA replication, si DNA polymerase 3, binabasa niya yung DNA template strand in what direction nga? Again, can you still remember? Binabasa niya yung template strand in the 3' to 5' direction. So that yung pag-synthesize niya ng complementary na bagong strand ng DNA is from 5' to 3' direction. Also, during transcription, binabasa ni RNA polymerase yung DNA template in the 3' to 5' direction so that yung pag-synthesize niya ng RNA strand is from 5' to 3' direction. So in summary, whenever we synthesize a new strand of nucleic acid, we build it in the 5' to 3' direction, and then we read the template in the 3' to 5' direction. But here, in translation, iba na yung direction. Si ribosome, hindi niya nababasahin si mRNA in the 5' to 3' direction. Instead, babasahin niya si mRNA in the 5' to 3' direction. So opposite na yung direksyon ng pagbasa ni ribosome kung ikukumpara sa direksyon ng pagbasa ni RNA polymerase at saka DNA polymerase 3. I hope you understand that. So that you can assume na yung start codon, yung AUG, nasa bandang 5' end siya ng mRNA. And then yung stop codon, wag uga ua, nasa bandang 3' end siya ng mRNA. I hope you get that. These are your codons right here. Again, when you say codons, they are found in the mRNA. And then the codons are complementary to the anticodons, or pwede rin the other way around. You say that the anticodons are complementary to the codons. The anticodons are found in the tRNA or transfer RNA. Take note, class, that whenever we write or read codons, it is already a convention, kumbaga common sense na, that we write or read codons in the 5' to 3' direction. So, going back to the start codon, yung AUG, it is always assumed that A is in the 5' end and the G in the AUG is in the 3' end. Yan yung convention, yung nakaugalian. Russell, Hertz, and Macmillan listed 5 features of the genetic code. But as far as translation is concerned, we are only going to talk about the 3 important characteristics of the genetic code. Number 1, and the most basic, the genetic code is a 3-letter code. Okay, I don't think kailangan ko pa siyang explain. Number 2, the genetic code has a start and stop signals. So, na-explain ko na rin yan kanina. Now, itong pangatlo, which is, ito yung i-explain ko talaga. The genetic code is degenerate. There is a phenomenon here called degeneracy or redundancy. So, from the word redundant, di ba? When I say redundant, parang sobra-sobra sa kailangan. Notice, class, na yung genetic code natin consists of 64 permutations. 61 of which are sense codons. So take note, 61 sense codons, tapos 'di ba 20 lang yung standard amino acids natin. Ibig sabihin, all these 61 will code for just 20 amino acids. So sobra-sobra yung number of permutations that exists, 'di ba? For example, at the most extreme, serine is coded by 6 codons in total. UCU, UCC, UCA, UCG, CGU, CGC, all these 6 codons code for just one amino acid, which is serine. Yung ibang mga amino acids dyan, kinocode ng tag-aapat or tagdadalawa ng mga codons. So, sobra-sobra. At this point naman, class, I want you to notice and appreciate na yung mga codons na pareho yung amino acids na kinocode nila ay magkakatabi sila sa table na nasa PowerPoint. Notice the brackets. For example, balik tayo sa serine. Yung apat na codon sa bandang taas that code for serine, yung UCU, UCC, UCA, and UCG. So, di ba naka-bracket sila? Notice na in all of these 4 codons, same yung first 2 nucleotides, U and then C. That means na as long as U and C, yung unang dalawang nucleotides ng codon, then it no longer matters kung ano yung pangatlong nucleotide. Hindi na importante yung identity ng pangatlong nucleotide. As long as U yung una, tapos C yung pangalawang nucleotide, then automatic, serine yung amino acid na kinocode niyan. Sa baba naman, yung sa baba na banda, yung sa serine pa rin, AGU, AGC. So, ibig sabihin, as long as A and G, yung first 2 nucleotides, tapos yung pangatlong nucleotide is either U or C. It doesn't matter kung U or C, basta isa sa kanilang dalawa, either U or C. Then automatic, serine yung i-code ng amino acids. Similar characteristic can also be observed in the other amino acids in the table that are coded by different codons. So question, how is it possible na minsan, in some amino acids, hindi na importante yung exact na identity ng pangatlong nucleotide as long as the same yung first 2 nucleotides? How is that possible? The answer to that is the wobble hypothesis put forth by Francis Crick. According to the wobble hypothesis, strict daw ang pairing ng first 2 nucleotides ng isang codon with the first 2 nucleotides of the corresponding anticodon in the tRNA. However, yung pangatlong nucleotide ng anticodon can wobble. Kumbaga, kaya niya magsayaw-sayaw. I hope you're getting my point. Basta, the point here is, hindi na siya masyadong strict sa kung ano yung nucleotide that corresponds to it in the codon. Kumbaga, for example, in the codon, yung pangatlong nucleotide mo is U. Diba supposedly, the partner of U is A. So, dapat A yung nasa anticodon. However, sa pangatlong posisyon, hindi na masyadong istrikto kung ano yung ipapartner mo sa A ng codon. It can be A again, or it can be U, it can also be G, it can also be C. Remember that sa partnership ng A at ng U nucleotides, ilang hydrogen bonds ang mabubuo? 2. However, in the wobble hypothesis, pinipermit na yung kahit isang hydrogen bond na lang ang mabubuo. Hindi na tayo stricto dyan. Now, what is the significance of this wobble hypothesis? Remember, you have 61 sense codons coding for 20 amino acids. Question, does the body or does the cell really have to produce 61 different tRNAs that will correspond to the 61 sense codons? The answer is no. Although there are 61 sense codons, but the cell does not have to produce 61 different tRNAs having 61 different anticodons. It can produce fewer than 61, and that saves energy and resources on the part of the cell that wants to conserve resources, di ba? That is practical, kumbaga. Now, another statement that I want you to understand by heart is the start codon defines the reading frame of an mRNA. For example, yung sequence of nucleotides in the mRNA sa taas banda That codes for the amino acids methionine, proline, glycine, glycine, threonine, valine, and glutamine. However daw, if nag-remove ka ng isang C na nucleotide, yung resulting amino acid sequence ay naging methionine, proline, glutamic acid, alanine, proline, serine, and asparagine. So we say na the reading frame changed because you removed one nucleotide. Similar effect will happen if you remove just two nucleotides. So the point being is the reading frame changes when you remove one or two nucleotides. But the reading frame will not change if you will remove three nucleotides na magkakatabi. Okay? Supposedly magkakatabi. Pag nagtanggal ka kasi ng tatlong nucleotides na magkakatabi, you simply uh, you simply removed an amino acid in the sequence, but the succeeding codons are not affected, and therefore the succeeding amino acids are not affected. So essentially, it's just like you removed one amino acid, but the reading frame did not change as long as the AUG serves as the defining part of the reading frame. So as long as na-recognize ni ribosome na ito yung AUG, then dito siya magsa-start, and that defines the reading frame. Okay, here is a short exercise for you, class. In the exam or in the quiz, you might be given a sequence of nucleotides in the mRNA, and you might be asked to translate that mRNA into an amino acid sequence. Alternatively, you might also be given as a nucleotide sequence in the DNA, and you will be asked to translate that. Pero of course, before you can translate that, i-transcribe mo muna siya into an mRNA, and then it is the mRNA that you will translate directly into an amino acid sequence. So I hope that you will be attentive to that. Okay? You have to be careful kung ano yung binigay sa inyo. DNA ba or mRNA? If it's DNA, then to produce an amino acid sequence, you have to transcribe that first before you proceed with translation. Also, isa sa mga challenge dito, class, is some students might be tempted to translate the mRNA into an amino acid sequence right then and then without remembering na dapat pala sa AUG sila magsa-start. Kasi ganun naman talaga in real life. Sa AUG nagsa-start. It doesn't mean na if CAG ang pinakaunang nucleotide sa mRNA, then that is the first nucleotide then that is the if CAG yung pinakaunang 3 nucleotide sa mRNA, then yan agad yung unang i-translate. Because remember, in the mRNA, you have this thing called 5' UTR, and you also have the 3' UTR. So in the 5' UTR, hindi yung pinakaunang group of 3 nucleotides yung tina-translate ka agad. But it will always be the AUG, which is the start codon. And then you have to stop when you encounter the UAA, UGA, or UAG. Okay? Basi ang uban sa inyo ha, class, kay magpadayon lang gyapon og kanang translate bisag bisag hagbay ra sila ning lapas sa stop codon. So you have to be careful with that. Ito, class, another concept that you have to understand. A polypeptide has directionality from its amino terminal to its carboxyl terminal end. I believe you are familiar with this because this is discussed in biochemistry. But ito lang, ulitin lang natin. If nucleotides have directionality, yes, they have their 3' end and the 5' end. So does proteins or polypeptides. Meron din silang directionality. In the amino terminal end, you can see the amino group of the amino acid that is free, meaning it is not participating in a peptide bond. And sa carboxyl terminal end naman, you can see the carboxyl group of the terminal amino acid. And that carboxyl group is also not participating in a peptide bond. Okay? Now, ito naman, look at the left side most part of the slide. So you can see there that the alanine, the amino acid carried by the tRNA in the picture, is attached to the 3' end of the tRNA. As early as na panghawakan ang mga fact na iyan, ha. Again, the amino acid, whatever amino acid that is, kahit hindi alanine, whatever amino acid that is, that amino acid is bonded to the tRNA in the 3' end of tRNA, not in the 5' end. Now, let's zoom in a little to see the molecular structure of the monomers of tRNA and also the molecular structure of the amino acid. So again, nasabi ko kanina, the amino acid carried by the tRNA is attached to the 3' end of the tRNA. And remember from our first lesson that in the 3' end of the nucleic acid, you can see a free hydroxyl group. Kasi ang functional group na naka-attach sa 3' carbon ng ribose or ng deoxyribose is hydroxyl group. Now, that hydroxyl group is the one that connects to the amino acid carried by the tRNA. So, again, tanungin ko, sa anong part ng tRNA nakakonect si amino acid? Sagot is 3' end. But if you will be more specific sa aling functional group at sa anong um, residue talaga naka-attach si amino acid, then the answer to that is sa hydroxyl group attached to the 3' carbon of the ribose ng tRNA. Ribose kasi RNA siya hindi siya DNA. So, hindi deoxyribose. Again, sa anong part ng tRNA naka-attach si amino acid? Sa hydroxyl group ng 3' carbon ng ribose ng tRNA. So, that's for the tRNA. Tanong naman, sa aling part ng amino acid nakakonect si tRNA? The answer to that is yung carboxyl group ng amino acid. Okay, ha? So, the carboxyl group, hindi yung amino group, hindi yung R group, hindi din yung hydrogen. The tRNA is connected to the carboxyl group of the amino acid. As early as now, panghawakan ang mga fact na iyan because that will be important mamaya and perhaps in our later discussions. Okay? Or perhaps when you go to medicine, mas magme-make sense ang mga fact na ito. As of now, they might sound trivial, pero importante din sila. Okay? So, since it is the carboxyl group of the amino acid that is connected to the tRNA, and then it is the hydroxyl group of tRNA that is connected to the amino acid, then what do you call the covalent bond between the tRNA and the amino acid? The answer is an ester bond. It is an ester bond. So later, pag sinali na natin sa discussion yung ribosomes, kung yung mga kaganapan sa loob ng ribosome during polypeptide synthesis, ito yung dapat na ma-imagine natin on the molecular level. Sa tRNA, balik tayo sa tRNA na may naka-attach na amino acid. Yung free end ng amino acid is the amino group. Tama? Tama. Kasi yung carboxyl group is attached to the tRNA. Therefore, the amino group or the amino terminal end is the one that is free. So, whenever na mag-a-add tayo ng panibagong amino acid sa growing polypeptide chain, it is the amino group of the new amino acid that is connected to the carboxyl group of the growing polypeptide chain. Uulitin ko ha, panghawakan ang mga fact na ito. Whenever na mag-i-introduce tayo ng panibagong amino acid during translation, yung amino group ng panibagong amino acid na yon is the part of that amino acid that reacts with the growing polypeptide chain. And yung part naman ng growing polypeptide chain that reacts with the newly introduced amino acid is the carboxyl group. Pakitingnan na lang yung diagram sa PowerPoint. Ito pa class, para mas magets nyo na. I am showing you now a sequence of amino acids and yung kaakibat niya na sequence sa mRNA. So again, the start codon is AUG. Tingnan sa baba, mayroong AUG. Yung corresponding amino acid niya is methionine. So, initially, yung carboxyl group ng methionine is the one that is attached to the 3' end of the tRNA for AUG or the tRNA for methionine. Now, imagine na-cleave yung ester bond sa pagitan ng methionine at sa kanyang carrier na tRNA. So, yung carboxyl group na yon is the one that will react with the amino group of serine the moment the serine carrying tRNA is introduced na naman to the to the ribosome. And then, imagine nyo, yung carboxyl group ng serine na nakabond sa 3' end ng tRNA will become free once the ester bond is hydrolyzed. So that the carboxyl group of serine now will be the one that will react to the amino group of the valine once a valine is introduced. And the process goes on and on. So that by the end of the process, the 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 ano ba? So that by the end of the process, yung amino acid na makikita mo sa amino terminal end is methionine and tinawag siyang amino terminal end. That is because the functional group of methionine that you can find there na malaya, hindi siya nagpa-participate sa any covalent bond, is amino group naman talaga. That's why tinawag siyang amino terminal end. And sa other end ng polypeptide, the free functional group that you can see is carboxyl group. Kaya nga tinawag yung end na yan na C-terminal end or carboxyl terminal end kasi yung functional group ng pinakalas na amino acid is carboxyl group. Okay? Okay. So notice in this diagram that pinakita dito, sinabi dito na the polypeptide is made from the N-terminal end to the C-terminal end. So again, it's not only the nucleic acid that exhibits directionality, but also the polypeptide. So, if binasa ni ribosome si mRNA in the 5' to 3' direction, synthesize niya naman si polypeptide in the N-terminal to C-terminal direction. Yun yung direction ng pag-synthesize ng polypeptide or amino acid chain. Again, synthesize si polypeptide in the N-terminal to the C-terminal direction. Okay, review naman sa mga past lessons natin. Synthesize si new DNA strand. In what direction nga? See it. Answer. The answer is 5' to 3' direction. In transcription, synthesize ni RNA polymerase si RNA. In what direction? 5' to 3' direction still. Okay, now let's review naman yung direction ng pagbasa. Si RNA polymerase, binasa niya si DNA template in what direction? 3' to 5' direction. DNA polymerase 3 read the DNA template strand in what direction? 3' to 5' direction again. But what about the ribosome? In what direction did the ribosome read the mRNA? In the 5' to 3' direction. I hope my repetitions helped you memorize the concept. We are already going to 38 minutes into this lecture video, but we still haven't really started the discussion on how translation really happens. All that we have covered thus far are the basic information that we have to memorize and understand, mga bagay na dapat pang hawakan before we tackle on the actual translation process. Okay? So now I'm giving you this table. I'm showing you this table. Please memorize na lang. Ang importante dito na i-memorize ay yung mga sedimentation coefficient na yan. Yung S na unit of measurement, that stands for Swedberg unit. That talks about the density or yung gaano sila kabilis mag-settle during the process that we call the ultracentrifugation. So you just read on that if you want to understand how that works. And I want to emphasize that the ribosomes of prokaryotes are different from the ribosomes of eukaryotes. They have different structures, they have different sedimentation coefficients, and it is important that you memorize these details. You will also need to memorize the different ribosomal RNAs that are present in each of those subunits. Okay? And now, finally, class, pag-uusapan na talaga natin ang translation. Out of my 69 slides, 18 lang talaga yung dedicated for the actual translation. The preceding slides are actually just for the preparation, not just to prepare you para mas maintindihan nyo talaga yung proseso ng translation. Hinimay-himay natin yung mga molecules involved in the process. So, just like transcription, which consisted of initiation, elongation, and termination, So does translation. Ganon din yung three stages niya. Initiation, elongation, and termination. And we're going to talk about all these three stages in a bit. And this is how translation is initiated. We begin with the small subunit of your ribosome. Associated to it is a tRNA that carries methionine. But in bacteria, methionine is modified. It is NF-met. Also take note that this tRNA is charged with one GTP molecule. Kakailanganin kasi yung GTP na yan later. Remember that the AUG codon, which is found in the mRNA, is read in the 5' to 3' direction, but the UAC anticodon, which is complementary to the AUG codon, is read from the 3' to 5' direction. I hope you are technical enough to remember these details. No? Also notice class that in this initial complex, the tRNA that carries the methionine is positioned in the P site. Okay? It is positioned in the P site. What happens next is that the initial complex that we formed later, that we formed kanina, it associates with an mRNA, which it will read. However, it will not translate, like I mentioned kanina, it will not translate the first three nucleotides, or essentially the first codon in the mRNA, because it will scan first the entire, no, not really the entire. It will scan first the mRNA until it encounters an AUG codon. And from that AUG codon, dyan na siya magsisimula sa pag-translate. Okay? So what do you call that region again of the mRNA that is not translated upstream from the start codon? The answer is 5' UTR, or the 5' untranslated region. At this point, class, I also want to point out, although hindi na natin siya i-discuss in greater detail, but I want to point out that methionine is methionine occurs not only sa pinakaunahan ng polypeptide sequence. Baka kasi isipin nyo na since methionine is the starting amino acid, then it is exclusively found in the N-terminal portion of the polypeptide. That is wrong. You can still find methionine somewhere in the middle of the polypeptide. It is because the AUG sequence, although it is the start codon, it is found not only sa unahan banda ng mRNA. Then the AUG codon occurs, or it can occur multiple times throughout the mRNA. In fact, class, there is a mechanism in which the very first AUG codon that is encountered by the ribosome is not actually the one that the ribosome will translate for the very first time. Meron din ganyan. Meron ding instance na ganyan. Although again, hindi na natin siya discuss in detail, yung mga mechanism underneath. So, yun lang. I just wanted to point out na it is possible na yung ribosome, naka-encounter siya ng AUG codon, pero iya hatong idedma. Dili to niya i-translate. Murag mangita siya og lahi pag yun na AUG codon. Okay? Possible na siya. So, kamo na lang magbasa ana sa inyong book if interested yun mo. Once the methionine-carrying tRNA is in the P site of the small ribosomal subunit, the large ribosomal subunit will now enter the picture. So, when asked in the quiz or in the exam, which ribosomal subunit first associates with the mRNA? Is it the large or the small subunit? The answer is small subunit. So, now, once the large ribosomal subunit becomes present na sa complex, it will now hydrolyze the GTP, which is carried by the methionine carrying tRNA, and then the initiation phase of translation is done, and we are now ready for elongation. For elongation, mayroon pa tayong dapat malaman. Si large ribosomal subunit ay mayroon siyang um, inherent na peptidyl transferase. Kumbaga, part na din siya ng large subunit. What happens next is, di ba bakante man kaganina ang A site ng ribosome. So there will be a new tRNA carrying with it its own amino acid that will enter the A site. kung unsa man tong amino acid and kung unsa man tong tRNA that will enter the A site, that is determined by the codon present in the A site, which is immediately next to the start codon. The peptidyl transferase will then cleave the ester bond between methionine and the 3' end of its carrier tRNA. Then, the same enzyme, peptidyl transferase, will form a new peptide bond between methionine, specifically the carboxyl group of methionine, and the amino group of the second amino acid. Next is yung pinakaunang tRNA that initially carried the methionine will now be present in the exit site. Then yung pangalawang tRNA carrying the second amino acid will now be present in the P site, and the A site is now empty and it is now available for occupancy by another tRNA carrying with it its own amino acid. And again, that tRNA which will occupy the A site is again determined by the codon present in the mRNA. By the way, hindi ko na-mention kanina, the process in which the ribosome passes along the mRNA is called translocation. Kasi 'di ba, ask yourself, paano ba yung pinakaunang tRNA carrying the methionine becomes present in the E site. Kanina nasa P site siya. Ngayon nasa E site na. Then yung tRNA carrying the second amino acid, initially nasa A site siya. Ngayon nasa P site na siya. Then later on sa E site na naman siya. So how did it happen? That is through the process called translocation, which is simply the movement of the ribosome along the mRNA. Translocation. And if napansin niyo sa diagram, mayroong mga GTP molecules na na-consume. Um, itong GTP kasi na to, kailangan to siya for translocation because translocation is an energy requiring process. Then the cycle will just go on and go on. It will just repeat and repeat until you end up having a very long sequence of amino acids, and that is your polypeptide. So paulit-ulit lang yun. Mag-translocate, may bagong tRNA na papasok, then mag-translocate, then yung peptidyl transferase, mag-cleave ng ester bond, and so on and so forth. Ganun lang yun. Paulit-ulit lang yun siya. Now, eventually, as the ribosome translocates along the mRNA, it will soon meet the stop codon. It's either UAG, UGA, or UAA. And that will be your termination. Look at the illustration. You now have a growing sequence of amino acids and your A site is vacant again. Supposedly, this A site will be occupied by a tRNA that carries with it its own amino acid. But since yung uwag o GAOAA kasi class, wala kasi tayong tRNA na mayroong anticodon na complementary sa uwag o GAOAA. Do you get that? For the 61 codons of the mRNA, mayroon yan silang corresponding tRNA. But for the uwag o GAOAA, walang tRNA na mayroong anticodon that is complementary to them. So, ano instead ang papasok sa A site? Imbis na tRNA, it will be an RF. RF stands for release factor. Now, hindi siguro nasabi sa book, pero nabasa sa book namin dati, if the tRNA carries an amino acid, what does RF carry? Ano yung baon niya? The answer there is water molecule, H2O. And how is that significant? Siyempre, kung wala namang baong amino acid yung release factor kasi water lang yung baon niya, so si peptidyl transferase, kanino niya idudugtong ngayon yung growing chain of amino acids? Sa water lang din. So, ibig sabihin, doon na magtatapos yung elongation ng sequence of amino acids. Hence, termination. After that, both the tRNA, which is now devoid of any amino acid, and also the release factor, and the large ribosomal subunit and the small ribosomal subunit will dissociate from the mRNA. And yun na yung termination natin. And here is a table for the summary of the comparison of bacterial and eukaryotic translation. There are terms here that we did not discuss, So if you are curious, just read on them in your book or panigurado lang for the exam, pakibasa na lang din. Just take it as part of your reading assignment. Yung mga Schneider-Gardner sequence, the Kozak sequence, yung mga different types ng release factor, the RF1, RF2, RF3, ERF1, and ERF3, yan sila ano na lang din, basa-basa, familiarize yourself na lang din para panigurado for the exam. But for our quiz, I will not ask them. We are almost done, class. Let us now talk about the polysomes or polyribosomes. Remember in our discussion on DNA replication, di ba, we mentioned the replisome and the replisome, aka replication machine, and there is also the replication factory. So, it is that the replisome is essentially an assemblage of all the proteins plus the DNA where DNA replication happens. Now, dito naman sa translation, parang mayroong counterpart yung replisome na yon. But ang tawag natin sa kanila is polysomes or polyribosome. What is unique about the translation process, class, is kanina kasi sa discussion natin, it is as if there is only one ribosome that translates the mRNA at a time. One ribosome at a time. But in reality, hindi ganyan kasimple yung mga bagay-bagay. In reality, there are multiple ribosomes working on the same mRNA such that the moment na natapos na si ribosome number 1 na mag-scan sa unahan na part, sa 5' part ng mRNA, let's say, nagsibog na siya, nag-translocate na siya, nano siya sa medyo later part ng mRNA. Since the 5' UTR of the mRNA is now vacant, then it is available for another na formation, na another initiation na naman. 'Di ba? Magbabind na naman ulit yung small ribosomal subunit, then the tRNA with a methionine, then katong large ribosomal subunit. I hope na ma-imagine niyo, class, basta ang point lang diri, class, is the moment na available na ang 5' end, medyo vacant na ang 5' end ng mRNA, kay nalampas naman dito ang nauna na ribosome, then another ribosome will form in the 5' end of the mRNA. So that daghang nga ribosome ang nagatrabaho sa isa lang ka mRNA, and you form this structure. Notice, class, sa diagram na sa 3' end ng mRNA, ang iyang amino acid sequence is medyo taas-taas na. That is because since naa naman sa 3' end ang kana siya na ribosome, tanawang diagram, class, di ba na ay ribosome ka na pinaka-last ka na nasa 3' end. Since naa naman siya sa later part ng mRNA, ibig sabihin hapit na siya mag-terminate. And since hapit naman siya mag-terminate, therefore, it only makes sense na ang amino acid sequence na gina-carry ng kana siya na ribosome is already long. In contrast, the ribosome na naa pa banda sa 5' end ng mRNA, wala pa siya or short pa lang ang iyang amino acid sequence. It is because bago lang manggod siya nag-start. So if you look at the entire polysome, you will appreciate that as you go from the 5' end to the 3' end of the mRNA, the length of the amino acid sequence increases. So, mura kag nagtan-aw og time lapse, good ana siya. And then ayan, an actual photo of a ribosome, of an mRNA rather, that is being translated by multiple ribosomes. The green colored structures that you can see are the amino acid sequences or the polypeptides. I just forgot the name of the technique. SEM man siguro ni. Anyway, the point here is you can see that the mRNA is ginatabangan siya ng multiple ribosomes. It's not just one single ribosome working on one mRNA. And that will be the end of our discussion for translation. I just decided to insert this picture here at the end part of my PowerPoint. This is another way of this is another representation of the genetic code. Yung kanina kasi na pinresent ko, naka-table siya, ito naman naka-circle. So maybe it is easier for you to memorize the genetic code in using this diagram. But then again, for the purpose of the exam and the quiz, you don't have to memorize the genetic code. Pero kung interested mo mag-memorize, you can do so. Lingawan po siya. These are your reading assignments. And yes, I am really expecting you to read on these things in your book based on your previous experience sa quiz 1 natin and sa quiz 2 natin, di ba? There were questions in the quiz na wala na ko siya i-discuss. So they were lifted from your reading assignments. So class, make it a habit really to read the book. Don't just read the PowerPoints or the trances. If naamay nagahimog trans sa inyong batch, you really read the book. Okay? And that would be all for this discussion. Thank you for listening. Thank you for your patience. And see you soon.